Hello, um, I'm now presenting a video about our paper, Sugarcane Bioenergy Research in Brazil, a bibliometric evaluation of the Bioen research program. The authors are Professor José da Silva Jr. from the University of Brasilia, myself, José Maria da Silveira from NIP Unicamp and Institute of Economics of Unicamp, Júlio César Carvalho Miranda from the Institute of Chemistry, Federal University of Mato Grosso, and Ronei Fraga Souza, the Department of Economy. To the next slide. The objective of the paper is to present a scientific production tied to the largest bioenergy research program carried out in Brazil in the period 2008-2019, the São Paulo State Research Foundation Bioenergy Research Program, Bioen FAPESP, using Bibliometrics and Scientometrics too. We just developed, alongside these years, our cooperation, scientific cooperation, trying to develop more sound analysis on not only in measuring the results of scientific and technological activities using papers and patents, but at some time try to interpret and use some tools to interpret better the results and to develop new index and new ways of reasoning about the impacts of scientific and technological activities. The features of Bioen program, I think that everybody of BBEST knows what is the Bioen program. I think that Bioen FAPESP is important to avoid. What is clear is the need, it avoids the thing that the dispersion of research activities. What I think that the research activities in many countries and many universities are located at the departments, and then there is some misincentive. If you only look for publication and your results, individual results, there's a disincentive to cooperation to develop more integrated approaches. I'm from economic area, and I think that the economic area is now being recognized as important to be tied up with other research fields in research and technological programs, because we know that's not only to perform, for example, cost-benefit analysis, but to go beyond, it's consider what would be the proximity of the technology to the markets and the possibilities of the technologies. And this is important. What the technology that is developed by a program or by a research group fit with the other technologies and fits the needs of the market. I think that it could be done alongside the research program, not only at the end of the research program. And then this kind of tools help us to locate it. What's going on in the university of the research that you are doing? You are doing research in some specific fields. What would be the links? What's the trends? What is the strength of the connections that you are doing? Okay, this is our methodology and data analysis. Okay, the data is impressive. I think that Bioen program has impressive results of research. And then we use only WOS data. There is much more. The problem of Brazil is that part of our research, our scientific communities focus on publishing in Brazilian, in some Brazilian journals that are not indexed in WOS. Okay, the final approach is community detection and identification of modularity. One can think that sometimes you can develop your own research in your own research field without any concern about what's going on in the other areas. Sometimes your results are very important and can be used by other researchers with any need of connection. Okay, you read the paper, you get the results. Okay, and then you follow the prescriptions or the patent indications, and then you include this knowledge or this technology advances in your research. This is modularity. But in other cases, when you do something, you open up connections with other areas. We believe that nowadays, connection is becoming increasingly important. So we try to identify situations that when you change, when you make a new discovery, you can influence. You have some, it works like genes, okay? Some epistasy effects that you have effects on the other areas that you can expect. You are not intentionally thinking about to do this impact in other areas. But if you identify this kind of clusters, you can think how in programs like Bioen that you can allow community to integrate some areas, not working separately. Okay, this is the main idea, is the subject of the rationality of Bioen in our approach. Okay, that there are some methodological aspects applying frontier methodologies in cytometrics. Okay, we have to build network. Now we're going further, constructing two-mode networks for international cooperation that allow us to combine, for example, for instance, countries and institutions. The great advantage of the network effect or network approach, that you can identify the importance of one single vertex. That means one single individual and the whole of the individual and the whole network. So you have the perspective, local perspective. You have more regional or more perspective. You are looking, for example, worldwide regions. Sometimes you have the whole perspective. So you can identify movings and the complete network, but you can identify movies in parts of the network that could be relevant for our analysis. I think that Bioen program contributes to a change in the perspective, the place that Brazil occupies in the international scenario of bioenergy research. Okay, we don't have this, it is not a final work, okay, it's a work in progress, but we believe that we can show with more econometric approach, more rigorous approach in the near future that Bioen perform this pattern. Okay, the data of Bioen are straightforward. You have almost 3,000 authors with 19 nationalities. It means that the program only spurs what was going on, but at some extent, the program is important to put people together. In the number of journals that are indexed in the Web of Science it's amazing relevant, and the number of citations, what is important, is becoming relevant. Okay, this is not easy to understand, but this is the yellow or orange line, I think, shows the dynamics of citation. You see that, for example, more recent papers are not allowed to have a lot of citations. This is a thing that we only can correct with time. If we perform the same research many years ago or many years further, we can see if the paper published in a specific year, for example, 2011, what the number of citations, what are the number of citations of the whole paper of Bioen program cited in a specific year, for example, 2011, and simultaneously we can see the average number of citations. So our results are it's in progress, because what we are seeing that is the speed of response of the program. We see that the program has impressive data, and there are some impressive response of the results. Okay, here we have the list of the most relevant journals that published papers of Bioen, and then the most important actors. Okay, the papers that more cited papers, and the number of cited papers you see in 2010, you have a paper with 770 citations made by Scuola of Mathematics and UFSCar is relevant. You have paper from NIPI, and you have the most important paper are from different institutions. What is interesting? This is a very complicated figure, but it's the most important, most relevant figure of our paper. It means that we can identify, for example, areas 4, 5, and areas 4, 5, and 6. That means biotechnology and applied microbiology. 5, engineering, biotechnology and applied microbiology. And biotechnology and applied microbiology focus on molecular biology, plant science. These areas are, there are 2 features. They're the most relevant. There are some extent you see by the links. They are integrated, and then they have important centers, leaders. Okay, there's a name for this now, it's a technical name in network analysis, but Policarpov, for example, Skinner, Maciel, Rubens Maciel, Vicentini, okay, are guys that you can identify are as a center of each cluster, but at same extent, these clusters are well connected with other clusters. And then when you see the figure on the agriculture, science, and technology, Marcus Buckeridge is a kind of cut. We say that if you skip Marcus Buckeridge from the network, all of the area of agriculture, science, technology will be disconnected from the rest. This is kind of cuts, a kind of bridges with the other areas. Marcus Buckeridge is, in this case, is an important bridge connecting this area with the main area that engineering, biotechnology, and applied microbiology. I think that the center position of engineering is another remarkable fact. Okay, here we have the main areas and the main activities by each country. What's interesting to know? What are the cooperation, for example, with Korea, material science, science and technology, chemistry. You can go further, can go to a more detailed sub-areas, and you have data for this. Here you have the importance, the links, the thickness of the links show the importance between different areas, different countries, the connection between them, and you see we call in our modern literature of complexity, what are the 2 centers and 2 peripheries areas. Okay, this is our similar what we have presented, and then this is a bipartite network showing the connection between countries and institutions. What are the more relevant institutions, what are the connections between them, and something we can see if the institutions are insular or they have a lot of connections with other countries. Thank you for your attention.
Hello, my name is Marcelo Cunha. I'm from Institute of Economics at Unicamp, the University of Campinas in Brazil. The title of my presentation is the Socioeconomic Impacts of RenovaBio policy in the Brazilian economy, described by a computable general equilibrium model. So, what's the RenovaBio? The RenovaBio is the new national biofuels policy in Brazil. It became a law in December in 2017. The purpose, the objective of our study, of our research, is to evaluate the socioeconomic as well the environmental impacts of this new policy in Brazil. So, what's the main idea of the RenovaBio? When the biofuel producer produces a volume of biofuel that is equivalent to reduce 1 ton of carbon dioxide equivalent emissions regarding fossil fuels, when this biofuel producer produces this volume is equivalent to 1 CBIO. A CBIO is a financial asset, and this asset, again, is equivalent of a biofuel volume that would reduce 1 ton of carbon dioxide equivalent emissions compared to fossil fuels. So the aim of RenovaBio is to reduce the carbon intensive fuel matrix in Brazil. So how to do that? So the Brazilian government has a target to reduce this carbon intensity, and then for that, the government can oblige a fuel distributor to buy a CBIO or a quantity of CBIOs, and when a fuel distributor buys a CBIO, as CBIO is attached to biofuel production, in reality, this fuel distributor needs to sell more biofuel and reduce the sales of fossil fuels. So when a fuel distributor buys CBIOs, these fuel distributors will increase the price of fossil fuels compared to biofuels, and of course, increasing the price of fossil fuels compared or against to biofuels, the consumers or the market, of course, will increase the consumption of biofuels and will decrease the consumption of fossil fuels. And of course, increasing biofuels consumption and reducing fossil fuels consumption, the carbon intensity of the fuel matrix will reduce. So in this slide, we can see the target of the Brazilian government to reduce carbon intensity. So when we compare the 2020 with 2029, in more 9 years, this reduction, the target is to reduce 11% of this carbon intensity. So to evaluate these socioeconomic and environmental impacts, we implement a general equilibrium model. And in this model for the Brazilian economy, we include the conditions of a RenovaBio policy in Brazil. So a general equilibrium model determines quantities and prices for all markets, for all products, in the sense to provide for all markets that supply is equal to demand. In this model, household consumption is consistent with the utility maximization, and the all primary factor by each sector is consistent with the assumption of profit maximization, of course, subject to technology constraints. So in the model that we have implemented, we provide a shock on carbon intensity fuel matrix. This is the objective, again, of the Brazilian government, and the model endogenously will determine what will be the price of CBIO, that with this price, this target of this carbon intensity can be reached. So considering the structure of the model, all variables and all parameters, the model endogenously decides to provide the price of carbon, the price of CBIO will be as well determine production changes, consumption changes, and price changes in all markets. And with those results, we can calculate the total impact on the Brazilian economy regarding GDP, inflation, energy consumption, carbon dioxide emissions, employment, and the land use change. So other assumptions that we assume in our model and the economy structure. So defined demand in our model is composed not only by by the household consumption, but as well government consumption, exportation, and investments. The biofuel sectors were modeled in the Brazilian economy with ethanol and biodiesel parameters, considering first generation. And finally, the share of household budget on fuel consumption was considered constant, but the consumption between ethanol and gasoline C can change according to their relative prices. Gasoline C is the blend uh, of pure gasoline. We say gasoline A and bioethanol. So to consider, I will return to this slide. To consider this this choice between ethanol and gasoline, we consider a curve that describes the behavior of a typical consumer, or the average consumer, or the representative consumer in Brazil, regarding to evaluate what will be the change in the volume of ethanol compared to the total volume in fuels when the relative prices between ethanol and gasoline changes. Of course, when this relative price of gasoline, of ethanol regarding to gasoline decrease, of course, the volume of ethanol will increase. So in the model, the CBIO production is attached to biofuel production, biofuel, ethanol, and biodiesel. The average fuel matrix, carbon intensity, takes into account 4 types of fuel consumed in the Brazilian economy. So bioethanol from sugarcane, pure gasoline or fossil gasoline, fossil diesel oil, and biodiesel. And we consider, in our case, that this biodiesel comes from soybean. So other assumptions: total biofuel markets is completely certificated. It means that all producers that produce biofuel, they can issue CBIOs. And other assumption is that the total fleet is flex fuel. So any light vehicle can choose, depending, of course, the relative prices, to consume ethanol, bioethanol, or gasoline. So in our model, we have 75 sectors, include all sectors regarding the fuel matrix. Yeah. Of course, taxes are included in our model. We consider three types of primary factors: capital, labor, and land. And our economy, of course, we consider an open economy, including imports and exports. So to evaluate the social, economic, and environmental impacts due to RenovaBio policy, we projected year by year the Brazilian economy from 2020 to 2029, and we took into account 3 scenarios. The first one is the Brazilian government projection of the economy, as well the fuel matrix. Yeah. In scenario A, the second one, we consider how would be the Brazilian economy without RenovaBio, and in this case, the price of CBIO is equal to 0. And the third scenario, scenario B, with RenovaBio policy, with the purpose to have to reach the target reduction in carbon intensity. And in this scenario, the model will calculate again endogenously what would be the price of CBIO to reach this scenario. So to evaluate, to assess the impact or the consequences of RenovaBio policy, we compare the scenario B against scenario A. That means we compare the scenario B with RenovaBio against the scenario without RenovaBio. So our main results. In this slide, we see that In green line, we see the impacts regarding the consumption changes in ethanol and pure gasoline with RenovaBio. And without RenovaBio, what we can see? We can see that the consumption of bioethanol would be 50%, around 50% of the consumption of bioethanol with RenovaBio, and without RenovaBio, the consumption of gasoline, fossil gasoline, would be the double of the consumption of gasoline, considering the policy or considering the conditions of the implementation of the RenovaBio policy. It's important to say that to these results, we consider that the international oil prices would be around $56 per barrel. So if without RenovaBio policy, Brazil would consume a lot of gasoline, pure gasoline, and the Brazilian economy would increase year by year the imports of gasoline. So with RenovaBio policy, we would see a strong reduction year by year in the imports of gasoline, reaching close 100% of reduction in the scenario of RenovaBio policy. And of course, in this condition, reducing close 100% of the gasoline imports, we would see an increase on GDP in the Brazilian economy. So regarding the consumption of the diesel mix, what we can see with the implementation of RenovaBio policy, we will see year by year a little increase in the total consumption of diesel mix. Diesel mix means the blend between diesel oil and biodiesel. And why, even with the biofuel policy, we see this short increase? The reason is that in the Brazil, the consumption of biodiesel is a mandatory policy. So with or without RenovaBio, we will not see a big difference with the consumption of diesel mix as well with biodiesel. So regarding the fuel prices, to reach this target of reducing 11% in carbon intensity in fuel matrix in Brazil, the reduction in price of ethanol need to be close to 15%, and as well, we would need an increase of 41% in the price of gasoline C. Regarding the diesel mix, we would see a short fuel price, even in 2029, less than 1%. And due to these changes in price in the whole economy, the impact on inflation in consumer price index, even in 2029, with higher reduction in carbon intensity, the inflation would reach more 4% when we more 0.40% when we compare the scenario with RenovaBio against without RenovaBio. So short impact on inflation, little impact. Regarding the CBIO's price, the model answered to us that CBIO's price need to increase year by year to increase the share of biofuels and to reduce carbon intensity. And in 2029, the price of one CBIO would reach close to 200 reais. Reais is a Brazilian currency, more or less $40 per CBIO. Yeah? In this slide, we can compare the answer of the model. For example, in 2029, our model answered that to reach this target of reduction of 11%, we would need 116 million CBIOs, and Brazilian government are considering that this necessity of CBIOs would be around 96 96 millions of CBIOs. So our model answered that would be necessary 20% more that is estimated by Brazilian government. In this slide, the main result is that when we compare scenario A without RenovaBio, yeah, considering the oil price $56 per barrel, we would see an increase on gasoline consumption, no change in ethanol consumption, all the time considering 2019 to 2020. So an increase of 0.6% in carbon intensity. So to reach this scenario of a reduction of 11% in carbon intensity, yeah, again, would be necessary to double the ethanol consumption, yeah, a decrease in 12% of gasoline consumption. The price of CBIO needs to be close to 200 reais per CBIO, or if the oil price would be, instead of $56 per barrel, $120 per barrel, in this case, the price of oil would increase so much the price of gasoline that would not be necessary to have a price for CBIO. Okay, so what's the very important conclusion until now? The conclusion is that RenovaBio in reality is a public policy for environmental hedge. So considering, of course, the biofuel production, we would see an impact on agricultural sector, mainly in sugarcane in Brazil. So with this policy, considering the hypothesis with the same land, the same area for agriculture production, we would see an increase in the productivity of sugarcane, but all other agriculture and livestock production. So finally, the main conclusions: the RenovaBio is an effective and necessary policy for the decarbonization of the Brazilian economy, promote the increase in biofuel use. So as long, of course, as the relative price of ethanol with gasoline C is reduced to the consumers. So CBIO's prices increases in unfavorable scenarios to biofuel and vice versa. RenovaBio should work, again, as an environmental hedge, since it protects the biofuel industry under unfavorable conditions. I mean, mainly due to high oil price scenarios. So, according to the simulated scenarios, the total value traded through CBIOs in 2029 would be close to 23 billion reais, close to $4 billion, and the actual CB target from the Brazilian government, as we saw, is underestimated. Finally, RenovaBio policy also has the potential to promote the improvement of agricultural productivity, while GDP, employment, and inflation didn't change significantly. So here you can see our contacts, yeah, my email and the mail of Carolina Habib Ribeiro that has worked as well in this research. Thank you very much for your attention.
Hello everyone, my name is Késia Lourenço. At the moment I'm doing a postdoc at Agronomic Institute of Campinas, and today I'm gonna present part of my PhD project, and the title of this presentation is Dynamics and Resilience of Soil Microbiome under Organic and Inorganic Disturbances in Sugarcane Fields. So this study is part of a bigger project called the Mini Bag, and the main goal was to uncover the main microbes related with greenhouse gas emission, with a focus on nitrous oxide production. Sugarcane is one of the most sustainable crops for bioethanol production in the world, because it can reduce the CO2 emission by replacing fossil fuels. And for Brazil, it's really important because in 2015, during the COP21, Brazil signed the agreement with another different countries to decrease the greenhouse gas emissions by 43%. So to achieve this goal, Brazil needed to duplicate the bioethanol production until 2030. Consequently, duplicate also the sugarcane production. However, we have negative effects as well with bioethanol production from sugarcane that is mainly related with nitrous oxide emission. Since 40% of the total greenhouse gas emission during ethanol production come from nitrous oxide production during sugarcane crop in the field. And also because during ethanol production, different organic residues can be produced and need to be recycled in an environmental sustainable way. The main residues produced are bagasse and vinasse. And the vinasse is the most important one because for each liter of ethanol, 10 to 50 liters of vinasse are generated. So in the last year, Brazil generated almost 381 billion liters of vinasse, and usually is used as organic fertilizer in the sugarcane field. Just for you to have an idea how huge is this amount, if you are going to storage the vinasse in Olympic pools, we are going to need actually 144,000 Olympic pools. And if you are going to apply in the field as organic fertilizer, using an average rate that you use here in Brazil, that is 100 cubic meters per hectare, we need to apply vinasse in the entire country, such as the Netherlands, even on top of the houses. And if you are going to compare with different states here in Brazil, we need to apply the entire state of Alagoas and Rio de Janeiro. So the amount is quite huge. And vinasse, as I said, is used as an organic fertilizer because it's a source of potassium. We can replace 100% of the potassium fertilizers. However, vinasse has also carbon and nitrogen, and this can affect the biogeochemical process, such as decomposition, respiration, nitrification, and denitrification. So this can change the microbial community. So how vinasse can impact the microbial community, the soil microbiome, in a short term, in a long term, and how is this dynamics in the soil? Also in taxonomy, in composition, but also in functions. How this residue can change? We already know that different disturbances can affect the soil microbial community in a different way. The community can be resistant. So when you apply a disturbance, the community don't change for a different state. The community can be resilient when the community changes to a different state. However, they come back to the previous one. The community can be functional redundant. So the community composition and the taxonomy changes. However, the functions are pretty much similar. Or the community just change in function and composition. So we need to know how vinasse and the mineral fertilizer can change the soil microbial community for bioethanol production to have a sustainable production. So based on this, we made different questions. One of them is the microbial community resistant or resilient to disturbances brought by vinasse application and inorganic fertilizer? Our hypotheses were the resident soil microbial community is not resistant to vinasse application. However, it's highly resilient. Vinasse applied a few weeks before mineral fertilizer will allow the community return to the pre-disturbance condition with less impact in the soil microbial community. The second question was how climatic conditions affect these responses of the microbial community to disturbances. Our hypothesis is that vinasse applied in the soil will change the community. However, the community will come back to the previous state, and after that, the environment conditions will be the drivers for this change in the soil. And the third question is there an interaction between fungal and bacterial community in the soil when we apply vinasse? So our hypothesis is that due to different lifestyle strategy, there is no correlation or no co-occurrence of bacteria and the fungal community. To answer these questions, we chose 4 different treatments. One is the control with only mineral fertilizer application, that's a common practice in Brazil. The second treatment is the application of a single vinasse at day 0. The third treatment is the application of vinasse at day 0 and the mineral fertilizer applied 30 days after vinasse application, that's the green treatment. And the fourth treatment is the application of vinasse together with mineral fertilizer in one single pulse disturbances. So we extract the DNA from the soil and also the DNA from vinasse because we know that vinasse has microbes and can be invasive microbes in the soil. So we amplified the 16S and the ETS to track these changes through time in the soil. And I forgot to tell you that we conducted this experiment for 389 days. So we collect soil through this time, 11 different time points. So here we have the main results for bacteria community. We can see here 9 different time points. And just to remember, the first vinasse was applied at day 0 and the second vinasse and the mineral fertilizer at day 30. So you can see that after 6 days after vinasse and mineral fertilizer application, the communities with vinasse and the mineral fertilizer are completely different from treatments with single mineral fertilizer and single vinasse. So the application of vinasse 30 days before mineral fertilizer did not decrease the change in the soil microbial community. Probably the carbon from vinasse was still in the soil, so when we applied mineral fertilizer, the microbes with copiotrophic lifestyle increased and changed the composition of the microbial community. And you can see that after 100 days, the community are pretty much similar. So the community is highly resilient. They come back to the previous state, and after that, the changes were related with environmental conditions. I must tell you that here in the southeast of Brazil, we have 2 different seasons, a dry season and a wet season. So completely different one from each other. Here to show the cyclical variation of the microbial community composition, we use the multivariate regression tree analysis. Here I chose to show you 2 treatments. The first one is the control with only mineral fertilizer application. We can see that after mineral application, the community changed and they come back to the previous state. However, after that, they were pretty much similar. For treatment with vinasse and mineral fertilizer, they are similar, with changes after both fertilizer application and after 100 days, the communities were similar. Related to the microbial invasion, the microbes from vinasse, they are all the microbes that comes with vinasse, they disappear from the soil after 30 days, with the exception of the Lactobacillus family. As you can see here in the control, that is the black one, there is no Lactobacillus family members here. However, in treatments with vinasse application, after 389 days, they start to growing back in the soil. Actually, you don't know they're growing in the soil, they are acylotic bacteria. And, well, they could survive there. So the main microbes related with the changes in the soil were microbes with copiotrophic lifestyles from Firmicutes, Proteobacteria, Actinobacteria. So the carbon from vinasse, they increase these microbes in the soil. Related to the fungal community, as you can see, the application of vinasse together with nitrogen or 30 days before nitrogen, they changed the soil microbial community. As you can see here, the treatments in green, blue, and red. There is high dispersion between time points. So they changed the community. However, the community is not resilient, such as the bacteria community. The community is not resistant. However, they do not come back to the previous state. Only the treatment with mineral fertilizer that they are clustered to really close to each other. However, after 183 days, the community changed. I must tell you that in this experiment, we had a high amount of straw on top of the soil. almost 60 tons per hectare. So probably here the environmental conditions and the decrease in the straw on top of the soil changed or shaped the community. So to show you the variation in the community, here I have the control treatments. Like see the previous figure, the changes were small. However, after 183 days, the community changed. And for treatments with vinasse plus mineral nitrogen, right after the application of the treatment, the community completely changed, and it was completely different from the other treatments. So the main microbes responsible for this difference were microbes that increased in treatments with vinasse, and they are related with carbon degradation. I must tell you that the alpha diversity that is related with the different species in the soil microbial community, they decrease with vinasse application. So these microbes here, they increased a lot in the soil, in the fungal communities, and there was a small number of fungal species in the soil when you apply vinasse. To answer the third question related to the interaction between bacteria and the fungal community, as we can already see that it's different. The patterns in the bacteria and the fungal community, they are different. The bacteria community is highly resilient. The fungal community is not resistant, but also not resilient. So there is no correlation between both communities. As you can see here, the correlation between the fungal families responsible for the differences in the soil microbial community, they are not correlated with the families that are responsible for the change in the bacteria community. So based on this, our conclusions are that vinasse is a source of microbes, nutrients, and organic matter. So these factors, they drove the change in the resident soil microbial community. And these changes were restricted to a short period, because the bacteria community, they have the capacity to return to the previous state. So the microbial community was not resistant to vinasse and mineral fertilizer application, but was highly resilient. And most of the invasive bacteria that comes with vinasse, they could not survive in the soil except the lactobacillus family. Related to the fungal community responded different to organic and mineral fertilizer. As you could see, that mineral fertilizer did not cause bigger changes in the soil microbial community. Disturbances changed soil fungal community without cyclical variation, so the community was not resilient. The community changed and is still different until 389 days after vinasse application, and the changes in the fungal community and the bacteria were not correlated. And the fungal community was not resistant and not resilient to disturbances. So I must say thank you to my group here in Brazil and the group from the Netherlands, and also to you. And if you have some questions about this work, just send me by email. Thank you. Bye.
Hello everybody. My name is Edson Wendland, and I'm going to present the work developed together by my colleagues from the Engineering School of São Carlos and from the Federal University of Mato Grosso do Sul. The title is Evaluating Hydrological Trade-offs due to different land covers and land use as fundamental input for assessment for ecosystem services. This work has been motivated by a recent publication in the Nature journal. It's called The Moment of Truth for the Cerrado Hotspot. As you know, Cerrado is the second largest biome in Brazil and used to cover almost 22% of the country's area. The problem is that we have a large expansion of agricultural activities over the Cerrado area, and this is the motivation for our study. So aims of the work are to evaluate the water fluxes in different LCLU types, wooded cerrado, also known as cerrado sensu stricto, sugarcane, pasture, and bare soil. We want to identify possible water balance trade-offs due to the different LCLU types. And finally, we want to understand the value and importance of watershed services as input for payment for ecosystem services program. It's experimental work developed in Brazil in Southeast region, and we see it in this general picture, the Cerrado area, and at the same time, our study area is located in an outcrop of the Guarani aquifer system. So we have two main reasons to develop this work. In an area close to São Carlos, we have a experimental site with two parts. Site 1, where we have different agricultural activities with experimental plot under pasture, sugarcane, and bare soil. And as a comparison, we have in the site 2, which is a remaining wooded cerrado area, where we want to understand the pristine conditions of the water cycle. In this sense, we have an instrumentation installed in the different sites. In the wooded cerrado, we have a tower with some meteorological devices. We have a deep well. We have sensor in the unsaturated zone. We measure the rainfall, true fall, and we also measure the interception in trees, and also the runoff and soil erosion. Here is how it looks like in the Cerrado area. We also have bare soil, where we measure only the runoff and erosion. Under sugarcane, we also have some sensor for the measurement of water and moisture in the unsaturated zone, and also true fall. And finally, in the pasture, we have installed a meteorological station, some devices for the unsaturated zone, and also a deep well to monitor the groundwater level variation. Here is how it looks like, the meteorological station, and also the tower in the Cerrado area. Our equation is actually to measure the soil water storage depending on the precipitation which is measured, the evapotranspiration estimated for different soil uses, and the surface runoff. The unit uses in millimeters per day, and for the soil erosion we use the unit of tons per hectare per year. This is the kind of results that we obtain. We have daily data as bars in the histogram, and we also have the cumulated, in this case, rainfall during the whole period. The second graphic is the evapotranspiration estimated for the different soil uses. Sugarcane in red, wooded cerrado in green, pasture in blue, and bare soil in gray. Also, daily data and the cumulated values along the time. And finally, we have in the third graphic the surface runoff, also in millimeters per day, following the same presentation. Daily values as bars and the cumulated values along the time. Using this data for our control period, we try to monitor the land use effects in the water cycle. So we use the wooded cerrado as the reference values and try to determine what are the changes promoted by agricultural activities. First, if we compare wooded cerrado and bare soil, we see a reduction in evapotranspiration of the order of 718 millimeters per year, an increase of runoff of the order of 134 millimeters per year, and a soil water storage in the amount of 584 millimeters per year. We also have the comparison of wooded cerrado and pasture, and here we see again a reduction in evapotranspiration, increase in runoff, and especially an increase in the soil water storage of around 570 millimeters. The values in the comparison between wooded cerrado and sugarcane are quite similar. Reduction in evapotranspiration, increase in runoff, and increase in soil water storage. We also compare the changes in the water cycle if we transform pasture areas in sugarcane plantations, and we see an increase in evapotranspiration of the order of 30 millimeters per year, reduction on the surface runoff around 29 millimeters per year, and also a small change in soil water storage. So we see if we move from pasture to sugarcane, the changes are not that large, but compared to the wooded cerrado, we have really significant values in the change of the water cycle. Since we have a significant change in soil water storage, we also have modification in the ground level variation along the time. Here we see the well in the Cerrado area, and in this graphic, in blue the positive soil water storage, and in red the negative soil water storage due to evapotranspiration from the Cerrado plants. Consequently, this is the curve observed for the groundwater level variation, which is very few because the soil water storage is very small. On the other hand, in the pasture area, where we also have a well, we see that the soil water storage, the balance of this equation, is mainly positive along this time of 5 years. Consequently, the groundwater level varies much more, and we can see the groundwater storage, the recharge, in the pasture, and compared to the Cerrado area, where we almost cannot see a groundwater variation. From this experimental work, the conclusions so far are: the water partitioning observation in different LC-LU types confirm that modifications in the land surface conditions may significantly change the water balance residual up to 584 millimeters per year, which we observe as soil water storage. The water balance outcomes evidence that the undisturbed Cerrado vegetation consumes the soil water storage in the dry season, which in our region occurs between June and September. That means the change in the soil water variation along the time is almost zero, and the evapotranspiration happens along the whole year because the Cerrado trees have a very deep root system and can extract water from the soil. On the other hand, the agricultural LC-LU, pasture and sugarcane reduce or even stop the water consumption in either seasons. So we have a very large positive soil water storage along the time, because the evapotranspiration almost stops during the dry season due to very shallow root system. We are extending our work, and for the future studies we have installed a flux tower where we try to measure the evapotranspiration over the Cerrado area to confirm the water balance, the residual. So we have installed a tower with 24 meters height above the Cerrado area, which has an average tall of 7 meters. Here is how it looks like. We see the tower installed in the remaining Cerrado area, and the instruments installed at different heights from the ground. We also want to monitor the litter production and interception in the Cerrado area. So we collect for some plots the produced litter and bring it to laboratory to quantify. We see how much litter is produced during the year. And then we developed a device which we call litter interception device to measure the moisture interception in the litter and also how much water flows through the litter and reach the soil. That's what we call LID. Is our device developed especially for this experiment. Here we see the lead installed in the field is a pluviometer. Above this pluviometer we have support for the litter which is installed, and all this is weighed along the time. So we see how much moisture is collected by this litter. We also have some ongoing research on environmental economics, and it's based at the detailed understanding of the physical process associated to the intensive use of land for meeting the water energy food nexus demand is of paramount importance in the context of sustainability. We have to understand what happens in order to quantify the environmental services, so that farmers, stakeholders and decision makers need to understand the value and importance of watershed services through a straightforward, cost-effective analysis of conserving and or protecting them. And finally, the economic feasibility affects the volunteer enrollment in payment for ecosystem service programs for adopting soil conservation practices in rural areas. So we are going to start a research with all the farmers and stakeholders to get more insight about their understanding about the environmental services and sustainability, especially in the case of the water cycle. I want to thank all these institutions that support our work: the Engineering School of São Carlos, University of São Paulo, the Graduate Program in Hydraulics and Sanitary Engineering, and the Arruda Botelho Institute, where our experiment is installed. We also thank the support by FAPESP, CNPq, and our partner in this research is the Embrapa company in Brazil. The main results presented in this short talk are published in this paper, Hydrological Trade-offs due to different land covers and land use in Brazilian Cerrado, developed by my colleagues as presented here in the authors list. I want to thank for this opportunity, and thank you very much for listening to our talk.
Hello, good morning, good afternoon, depending on which part of the world you are right now. My name is Marcelo Moreira, and I'm going to share a little bit with you the research we have been performing regarding second crop ethanol in the context of advanced biofuels. But first, I'd like to thank the organizers of this session and the organizers of the BBEST 2020-2021 Biofuture Summit Conference. About Agroecony, our company is a company dedicated to provide knowledge for a better economy. We work closely to the agricultural sector, but also with land use. We do work with global agricultural analysis, applied modeling, sustainability and environmental regulations, communication strategies, and territorial intelligence. We have worked in all the most relevant policies in the world for biofuels, including US EPA, Renewable Fuel Standard, Low Carbon Fuel Standard, European Commission, the Renewable Energy Directive from 2010 to 2020, and most recently the RED 2, which is between 2020 and 2030. We also have been invited to join the technical group in the design of the RenovaBio program. We are very proud of that, as well as we are also part of the COSIA certification scheme, which covers biofuels for international aviation. So a little bit of context regarding this research. It was in 2017 that we heard about intentions of investing in a new corn ethanol facility in Brazil. Corn ethanol was already very well known in the United States, but it was heavily dependent on fossil fuel for heat. And Brazil has always tried to distinguish Brazilian ethanol, which was based on sugarcane, and the ethanol produced in the United States. Some feature regarding this investment: it wouldn't be the first investment in corn ethanol in Brazil. There were already some mills existing at the time, but it would be the first mill in Brazil that was going to produce ethanol 100% from corn. You can see here a map from Brazil, right? Those are the biomes for Brazil, the Amazons around here, Cerrado, Caatinga, Atlantic Forest, and here we have the state of Mato Grosso. The state of Mato Grosso has a particular production system, particularly for corn, where the state only produces corn from second crop. Okay, this is a map of the state of Mato Grosso with the facilities of corn ethanol within the state. We have taken as reference one facility that was going to be built. This was the first one, what we call full facility on corn ethanol. It's on Lucas do Rio Verde. It was going to produce at the start 250 million liters of ethanol per year, and then it has expanded to 500 million liters of ethanol per year. As you can see here in the map of Mato Grosso, we have different levels of production of second crop of corn, and what you see in brown, dark brown, is where you have most of the production. So you can see that Lucas do Rio Verde is placed on the place of the state where we have much of the production of second crop of corn. And then here we have the production system. So for each ton of corn, we can produce, this is quite similar to the production part in the United States. For each ton of corn, you can produce about 430 liters of ethanol that goes to the transport sector, 363 kilos of DDG that goes to the feed sector, and a bit of corn oil, which can go either to biodiesel production or also to the feed sector. However, to produce such ethanol, energy is needed, and this energy comes in Brazil from a CHP plant, combined heat and power, which is fueled by eucalyptus chips. Right, so biomass is the main source of energy to run the facility. It is interesting in this case that all the energy that is consumed in the ethanol facility is lower than the CHP plant can produce. So the excess of electricity that is not consumed in the ethanol plant is exported to the national grid. Here we have a very important feature of the plant in Brazil, which is this production based on second crop of corn. Here we have a picture of an operation of second crop of corn. Here you have the harvesters harvesting soybeans, and then they are planting corn at the same time. Basically, soybeans used to be produced in a longer time, but several management practices were applied, and the soybean growth cycle has been reduced. So now we can produce both soybeans and corn within one single year, right? Some numbers here. In last decade, the production of corn increased 51 million tons, so almost double, and the area of first crop of corn has reduced by almost half, 43%, not almost half, but you can see the big numbers. And this is because the area of second crop of corn has been growing, where the area of first crop of corn is declining. The first set of results refers to the attributional approach, carbon footprint of the corn ethanol in Brazil on an attribution approach. You can see that results, the carbon footprint is quite low, 25.9 grams of CO2 per megajoule in one way to model, same system, but in the same system, different way to model, resulting into 18.3 grams of CO2 per megajoule. We can see that the bulk of the emissions are related to maize production, between 20 and 12%, and here the difference between one and the other is just the way we have treated the emissions related to the agricultural phase. Such an impact is due to the co-production of soy and corn, and of course how we deal with this. But anyway, the results show a very important reduction compared to gasoline. Gasoline is expected to be 87 grams of CO2 per megajoule in Brazil, so it could be around here, and this represents a 79% reduction between gasoline and ethanol from corn. And here we have a comparison of our results in the right with the existing results so far for corn ethanol in the US. As you can see, so we have a very different profile of emissions, where in Brazil most of the emissions are related to maize production, and we have very small emissions in the processing. In the United States, we have significant emissions on processing, so it goes from 29, 40, 41, 51 grams of CO2 per megajoule in the United States in the processing or fuel production and transportation, which is basically the emissions at the mill and transportation. This is because in the United States, a lot of the energy used for processing the corn and transforming it into ethanol comes from fossil sources, for example, natural gas. In Brazil, this is much smaller. Another difference that you can see in this chart is that we have here the land use change emissions, which can be considered to be important in international discussion. It goes from 30 grams of CO2 for the first version of the low carbon fuel standard, and it has been reduced to 20 grams also in the low carbon fuel standard. So this is the current carbon footprint, land use emission carbon footprint in the low carbon fuel standard. And this is 26 grams of CO2 per megajoule in the renewable fuel standard. So it's important to investigate that as well for Brazil. And this is the big surprise, most exciting result of this research. We had a negative emissions regarding to land use. So what explains the negative emissions are the expansion of eucalyptus into cropland and pasture, but also the displacement of soybean meal and corn for on the feed market. So the DDG displaces corn, soybean meal, and corn. So you need less land to produce the same amount of feed. We have also performed other tests, for example, with no expansion from eucalyptus, which delivers less credits, I would say. Lower DDG nutrition capacity, which would be a worse scenario compared to the one we have. But we can also have higher nutritional capacity of DDG, and then the result would be better compared to what we have right now. This is the results for the consequential LCA. So this is land use that we have just shown to you. And also here we incorporate the credits for electricity export when bioelectricity is considered to displace the marginal electricity source at the Brazilian national electricity grid. So all the emissions goes up to 20 grams of CO2 per megajoule, but we also have those credits, and the final score is a carbon footprint of 4.5 grams of CO2 per megajoule, which is quite low. Depending on the alternative scenario, this can go up to 9.8, 7.6, but it can be as low as 0.8 using current technology. So it's basically zero, which is quite relevant in terms of indication for policies. Here a comparison again with corn ethanol in the United States. We also have the same information as before, so most of the emissions are related to corn, and small quantities are related to the ethanol production, but we do have credits here, when in the United States we don't consider such credits for electricity or for land use. So this is quite different, and of course this overall results quite different, from 4.5 to 0 compared to the best case scenario is 52 in this result here. And one very relevant one is the current carbon footprint in carb, which is about 78 grams of CO2 per megajoule. So we can say that corn ethanol in Brazil is quite different from corn ethanol in the United States. As for final considerations, it's important to consider that corn ethanol is here for good. It started the production with 0.2 billion liters per year. That was the first facility, and the whole sector currently produces 3.5 billion liters per year. This facility that we have studied also has finished his evaluation on the RenovaBio program. It got the best score for anhydrous ethanol among 220 ethanol producing facilities in Brazil. So this confirms the result we have received, and it also had the second best score, and then this is for anhydrous ethanol. What is really interesting as well, we all know how important it is to have negative emissions, and particularly in the energy sector. The company that we have studied is now studying carbon sequestration and storage, which would lead them to be a negative emission producer in the energy sector. And here you have my contacts. Please don't hesitate to contact me should you have any doubts. Thank you for listening. Bye-bye.